my major push here is to really encourage you all to get engaged with the research effort in FSH. The more people that are involved, the more we can do, the faster we can move things forward. And involved does not mean, anything terribly onerous or time-consuming or energy-consuming. If you literally just make sure you're connected with the society, make sure you're connected up with the National Registry through the University of Rochester, and get connected with any centers that are doing research that you would consider going to participate in a study. That's huge. That's really of value. We recently completed a study of a novel anti-inflammatory drug on FSH, and it was really a struggle. We had trouble getting enough patients in to really get the study done. And so this is the kind of thing where we really need to partner, link arms with the research community and the patient community so we can really do more. And the most important thing is this is an incredibly exciting time. I mean, Howard was talking about this earlier, but I just have to tell you, and I'm going to end on a note demonstrating that. So things are moving forward, and we want to move them forward as fast as possible. So what do we need for a clinical trial? Well, we need a proposed treatment, but we need an affected group of patients, by which I mean access and patient subjects that we can actually work with. We need an understanding of the effect of the treatment. We need to know what we're looking for, and we need to understand the natural changes that would go on in a patient without the treatment, so we can see whether or not the treatment is actually responsible for any effects. And then we have to measure it, and we have to prove that it's acting biologically the way we want, that it's having clinical benefit. If we correct something, that we can show that there's a genetic correction and everything looks great in the microscope or in the DNA assessments, that will not get a drug approved unless it actually is shown to make people better. You have to live longer, you have to be stronger, be able to do more, something. There has to be an actual clinical benefit. And then we have to prove that it doesn't cause a lot of trouble. So what are the phases of clinical trials? We start with natural history studies, sometimes called phase 0 studies, where we're just monitoring patients, seeing how their condition progresses without treatment. That is an essential part of this, because that allows us to know what we're going to look for when we start using a treatment. Those studies tend to cost on the order, depending on what assessments we're doing, of $1 to $10 million. The point here is that these things aren't cheap, and for us to get, first off, the NIH and funding organizations to spend money, and then to get pharmaceutical companies to spend money on these things, it's to all of our benefit if we can show that we can get these studies done, lickety-split. So great, we've got a new idea, we start a study, boom, we get it done within a year, we get it done within 2 years, and we publish it and move on. That attracts more attention to the field. That allows us to do even more. So that's where we want this to cycle in a positive way. Okay, so we've got a treatment that looks good in the laboratory, and we've got a natural history that we think we can impact. First thing we do is rule out toxicity. That can be done fairly quickly. This falls into the responsibility of the drug company, costs $10 million. We move to the next phase study. Each of these takes between 6 and 12 or 18 months to complete. So you can begin to add up the time here. The phase 2 study will cost $10 to $30 million commonly for us to get these things done. They get to be quite expensive, and when we come up to doing a phase 3 trial, which is the level of trial that we need to present to the FDA to show that we really have an effective drug, we're talking at least in total about $100 million. So you're not going to be doing a lot of these because nobody has that much money available unless it really looks promising, both from a biological standpoint and what they're seeing in the lab, and from a clinical standpoint, that we can get the study done and that we can do it in a reliable way that's going to give us an answer. Drug companies don't care as much about doing a study that doesn't give them the right answer as they do about doing a study that gives them no answer. That will really scotch this. They will not want to continue working with us if we can't get the studies done and done in a timely way and in a way that really is interpretable. So what defines a good clinical trial? We have to have the right population of patients. I'm going to talk a little bit more about that. People have to be affected, but not too affected. We have to think we're going to see an effect of the drug that we're studying. We have to have enough of that type of patient to do the study, and then we have to be able to complete it, meaning those patients have to be around and able to participate throughout the study. We have to investigate some feature of the condition that we think is going to respond to treatment. And I'm being somewhat vague, but it's inevitable. We don't just give a drug to someone with FSH and have them give a global score of, are you better? I mean, we have to have something we can measure that either gets better or that without the treatment would have gotten worse. And so we have to have something, again, this comes back to the measurability of this. And so we have to have then an appropriate measure that everybody agrees we're doing it in an objective way, we're doing it in a reliable way, and in a reproducible way. This then we can present to the FDA, and the FDA will go, yep, we know those guys, you did it right, beautiful data, everything looks good, fine, the drug gets approved. The goal here is to get drugs approved, not just to do studies, but to do studies, we need to do the studies to get the drugs approved. Very few people are going to benefit from the drug in a trial, but once we get it approved, thousands of people can benefit from it. So there's reason to be excited about this. It's all moving in the right direction. So I thought I'd try to put this in graphic form so you could get a sense of this. And it's funny, I came up with a curve that's similar to the one Richard showed. So if we look at strength in an unaffected individual, they start weak at birth and then get stronger, and then their development of their strength has matured. They've gotten to their full development. But there's also an aging process that needs to be added on to that. So it's aging or degeneration, and that then will be superimposed on the developmental curve. And then on top of that, there's going to be some fluctuation. So we all know that some days were better than other days, some years were better than other years. And that can be due to sleep or stamina or kind of all kinds of different things. And when you put these all together, you get a composite curve for some typical person that's unaffected by a neuromuscular disorder. So that would be roughly the way you would expect their strength would appear over time. In someone with FSH, it's more complex. We think there is actually an effect of FSH on muscle development. And so their muscle might not quite develop as rapidly, it might not develop as fully, and they might have some modest degree of weakness. Now, I want to point out that this isn't generalized. We talked earlier about the pattern of muscle involvement. So some muscles might be fully normal, but other muscles might not be, even early on. But it's not apparent unless and until that muscle begins to actually age or degenerate, which is also an altered curve in FSH, so that the age at which they begin to show signs of muscle deterioration might be earlier, the rate at which they begin to deteriorate might be faster, and they also might have greater fluctuation because they have more systemic involvement, more pain, and other things. So you add that together, and they have a different curve over their lifetime of what their strength looks like. And again, you can think of this in global strength, doing things like walking, how far can they walk, or you can think of it in terms of individual muscles, how an individual muscle functions. So these are just schematic, these are just concepts. So if we want to then measure strength over time in an individual, it's going to be a noisy curve. It's not a perfect smooth curve. It's variable from day to day, week to week, year to year, but it's going to follow some kind of a course like this, where it gets better for a period of years, decades, and then maxes out and then begins to gradually change. What we want to do when we do a clinical trial is we measure the function between two different time points. So if we measure in this instance, if we take a child, say, and measure their function, this again can be walking or it can be bicep strength or whatever we're looking at, we might see in this instance that between time A and time B, they actually got stronger. So if we had studied this person and given them a drug at time A, we would say that drug worked. You could give them chicken soup and it would look like they got better, right? Because that was their natural curve, was to improve. Alternatively, if we studied somebody at a different time and we gave them their drug at that time, we might say they got worse, even if the drug worked somewhat, because their natural course is to get worse. Or if we looked even later, it might be the best drug in the world. It might be a phenomenally effective drug at a different age, but if you're studying it in the wrong population at a time when they're no longer apt to respond, you don't see any effect at all. So the point here is that you have to choose the individuals that you're studying and compare with and without in that particular time group. The other way you do this is to compare more than one person, obviously. But that's a problem too. I mean, if you compared the person on the left, if you added the person on the left and the person on the right together, you have one person that's naturally getting better and one person that's naturally getting worse, and everything cancels out and you don't get anything. So the point here is that the natural history, understanding what is apt to go on, is going to allow us to identify the correct group in which to do our study. We're trying to develop a drug that's applicable to everybody with FSH, but we're going to refine our study to a certain window just so we can get the study done. If we include everybody with FSH, we're going to kill ourselves. We're going to undercut the point of the study. And that's why we restrict the criteria to get into these studies, so that we can be comparing people that are very similar. We know what's going to happen without the drug, and we see what happens with the drug, and we compare groups that do get the drug or don't get the drug. The more we can limit variability, the more information we can get out of this. So that's the name of the game. So how do we choose that right study population? All the patients should need to be similarly affected, and all of them should be expected to progress in the same way. We could choose a person that has the same strength and is expected to get better and compare them to a person that's got the same strength and appears and is predicted to get worse. So if we just restricted who gets into the trial by how strong you are, and we didn't anticipate which direction their natural curve would go, we would be screwing up the study. So you really have to know a lot about what you expect to happen in order to really refine this. And there are countless stories. I could give you lots of stories of studies that have failed the drug, where we have a drug that looks like it works. It's not that the drug fails the study, it's that the study fails the drug. And that we don't know enough, we don't do it right, and we keep on learning. And this is where I come back to this. The pharmaceutical companies, as long as we're learning something, might come back and say, oh crap, we just spent $100 million and you screwed it up, we got a crappy result. But you really informed us about how this works. Now we know we can go back, we can do this again with a slight difference in the way we design the study, with the chance of actually doing this. So we've had pharmaceutical companies go through 3 phase 3 studies, so hundreds of millions of dollars applied to getting these things right. And we don't want to do that. We want to get it right from the get-go. But that's the name of the game. All the people need to be able to complete the study, as I mentioned, that we can talk about more. And we have to have enough people so that we can actually power the study, meaning that the effect that we expect to see from the drug is going to be greater than the affected change in progression and fluctuation, so that it comes out of the noise level. If we do 20 people on the drug and 20 people without the drug, or however many we need to. So we calculate how many people we need to see involved. As you might imagine, if we want to restrict the subjects, so we say we want people with FSH who are 20 years old and can move their arms only so much, but not too much, but this, and we really define that precisely, we limit the number of patients and subjects that can participate. So we end up shooting ourselves in the foot. We can't get the study done because we've defined the criteria too narrowly. And so it's always a compromise, and that's why this gets to be a challenge. So what's been developing for FSH? Gene manipulation, altering FSH in a number of different ways. There's a lot going on. You've mentioned it. We'll talk a little bit more in terms of the Acceleron study, which is very exciting. What can you do? You can register with the FSH Society, you can register with the National Registry, and you can register with Stanford. Just Google Stanford Neuromuscular Research and you can find contact information, or other sites that are doing research. And I really am eager to work with you and the FSH Society. I just wanted to show, I'm going to go 2 minutes long, to show you the results of a different disease that we've studied on SMA. And the reason we studied SMA is because we can be really precise in the type of patients we see, so that we know how old they are, we know exactly what's going to happen. It's a god-awful disease. These little infants die in about a year or 2 years of life, and but we can identify them before they're severely affected, and we can see enough of them to get the studies done. And the reason I bring it up is because this drug was just approved by the FDA, and it's revolutionizing this condition. We can now treat it. These are the graphs of the results, which I don't have time to go over, but I did want to at least show you a couple of videos. So this is the very first infant with SMA ever treated. And this is before she was treated. You can see how weak she already is. She can't move her legs. She can twitch her toes. She can move her arms from the elbows down to the hands. Her parents are going to sit her up here just to take a picture, but she can't sit up long because she's already losing the ability to breathe. Without treatment, she would have undoubtedly died within a matter of months, or would have had to have been put on a ventilator to keep her alive. She was treated now 4 years ago, and here she is now. So it's not a magic wand. I mean, she's still got a disability, but she can stand up in her stander and play the piano and zip around in her power wheelchair and breathe on her own and go to school and yell at her baby sister and do all kinds of things. I bring it up because I want you to understand this is the technology that allowed us to do this is the same technology that we're using for FSH to genetically correct FSH. It's in earlier stage in FSH, but working together, if we link arms and work with the community, we can do this. So I am more excited than I've ever been in the decades I've been in this field. And I wanted to share that with you and to let you know that we can do a lot working together. So thanks.